Hello and welcome to Plant Breeding Genomics webinar series. Today we're going to be meeting with Fikret Isik on genomic relationships and the GBLUP. He is an associate professor at North Carolina State University, and I am Sean Yarnes, who'll be hosting the webinar and will be directing the question and answer session at the end. And so there's a chat box you'll notice on the webinar control panel, and as you think of questions for Fikret, just go ahead and type them in the chat box and I will read them to him at the end. You can find all of our materials for plant breeding genomics on eExtension at www.extension.org/plant_breeding_genomics. And next week we'll have recordings of this broadcast up on the internet, as well as all of Fikret's data files and script files. So I'll be sending out reminders via Facebook, email, newsletters, and so forth. But you can check back in a week if you'd like to see the recording. So now I'm going to hand things over to Fikret. Enjoy the webinar.
Thank you, Sean. Okay, I have 30 slides out of 44 I will cover. 3 slides about introduction, 6 slides about matrices needed to calculate the genomic relationship, and 8 slides about methods to calculate genomic relationships from markers. And here we'll have a demonstration using R. And then we will review ABLUP, which is the traditional genetic evaluation using the pedigrees and the phenotype. Then I have 5 slides about genomic BLUP. If time permits, we will have another demo to run GBLUP using ASReml. And then I have 3 slides in the conclusions to show some results from one of the manuscripts came out very recently. Okay. So traditionally, breeders have been using average genetic relationships and phenotype to calculate the breeding values. The genetic relationships between individuals derived from pedigrees are discrete. For example, we assume that full SIPs share about 50% of the alleles that are identical by descent, and half SIPs share about 25%. But we can use markers actually to be, more precise about the amount of DNA shared by the individuals. And this was first proposed by Van Reden in 2008. Markers actually estimate proportion of chromosome segments shared by the individuals, and including the genes identical by state, not just identical by descent. So before we, we launch on about calculation of genomic relationships from the markers, let's review some basic information about the markers and marker matrices we need to calculate genomic relationship matrix. Genotypes and gene content. I'm sure you are familiar with those words. Let's say we have 3 diploid individuals and 4 loci. And the lowercase letters represent the minor alleles at each locus. And the capital letters represent the major allele. So we have a matrix of the genotypes, 3 individuals, 4 loci. So the genotypes above can be converted to gene content, which is basically the counts of minor allele at each locus. And that calculation is shown in the matrix below in the slides. And let's call it, MAF matrix, or minor allele frequency matrix. So if we look at those elements, individual 1 in locus 1 or SNP1 has 0 minor allele. And individual 3 in the same locus, locus 1, has 2 minor alleles. So one of the matrices we need to calculate genomic relationship matrix is the M matrix. This is basically we take the deviations of 1 from the gene content, and then we obtain 1, 0, or -1 for ease of subsequent calculations. So when you have large matrix, like 40, 70,000 of markers, it can be sometimes time-consuming to make calculations. That's why statisticians, convert a minor allele frequency matrix to M matrix, and the elements are 1 or 0 basically, so it's easier to calculate. And on the right of the screen, you can see the R script to do that. It's very simple calculation, of course. So with the data formatted like this, now we are ready to compute a matrix of realized genetic similarities among all pairs of individuals. But before we do that, let's look at the product of the M matrix with its transpose. And those are interesting to look at those elements. In the diagonal elements, we see the counts the number of homozygous loci for each individual. So those are like blue highlighted numbers. For example, first individual has 2 homozygous, and second individual has 2 homozygous, and third individual has 4 homozygous loci. Off-diagonal elements are interesting too. They measure the number of alleles shared by the relatives. We can multiply the M matrix with the transpose by reversing the order. As you know, changing the orders of matrix multiplication gives different outcome. So in that case, we have diagonals representing actually the counts the number of homozygous loci for each locus. For example, locus 1, which is SNP1, has 3 homozygous individuals. So going through the diagonal elements, you can tell, how many homozygous individuals each locus has. And off-diagonal elements, they measure the number of times alleles at different loci were inherited by the same individual. So, taking the transpose of the M matrix and looking at the products of the M and M transpose gives us different information about the data. It's very informative. So another matrix we need is P matrix. The columns of the P matrix are allele frequencies expressed as a deviation from 0.5 and multiplied by 2. So let's say the minor allele frequency of 4 loci are given P sub 1, 0.383, and then other, minor allele frequencies for given loci are given here. So we can use those minor allele frequencies, P sub 1, P sub 2, P sub 3, and P sub 4, and then we can calculate the elements of the P matrix. It's a very simple matrix algebra, and the result is given at the bottom, the P matrix. So once we obtain the P matrix, we can take the subtraction from the M matrix and obtain the Z matrix. So Z matrix is one of the major matrices we use to calculate genomic relationship. What we are doing here is that we set the means values of the alleles effects to 0. So by subtraction the P from the M, we are giving more credit to rare alleles than to common alleles when calculating genomic relationships. Because really, rare alleles matter more than the common alleles when we look at the relationships between individuals. Genomic inbreeding coefficients is greater if the individual is homozygous for rare alleles than if homozygous for common alleles. So we get different inbreeding coefficient if we have rare alleles compared to common alleles. So we covered different matrices derived from the markers to calculate genomic relationships. Now let's look at some of the methods suggested in the literature to calculate genomic relationships. The first method, I number them from 1 to 5. There are more than 5 actually, but I am only covering 5 or 6 of them. This is called GOF matrix. It's derived from the observed allele frequencies. So we know what Z is. It's the incidence matrix for the marks we just talked about. And the denominator in the formula scales the G to be similar to the A matrix. And P sub I are the observed minor allele frequency of all the genotype individuals, regardless of inbreeding and selection. So this very simple formula, ZZ prime is weighted by the minor allele frequency of observed genotypes. Another way of calculation of genomic relationships is GD matrix. This is variation of the previous matrix, basically. The difference is, weighing the ZZ prime matrix. So instead of using a different denominator, we are using reciprocals of markers' expected variances. So D is a diagonal matrix. In the diagonal, we have elements of markers' expected variance. So between 1 and 2, the difference is weighting, weighting the ZZ prime. And then GO5, when minor allele frequency in the base population is unknown, then 0.5 is used for all the values of the P sub I. And another one is GMF. Again, it's about the minor allele frequency in the base population. When it is not known, we use average minor allele frequency of genotype population to calculate the minor allele frequency. So it depends on what kind of minor allele frequency is used, matrices are named GO5, GMF. Regression method, I call this matrix as Greg. So NM prime is the dependent variable, and A matrix is the independent variable. Here, the objective is to calculate the intercept and the slope. So we want to actually get the solution for A using the markers as the dependent variable. So the error actually includes the difference of true from expected fraction of DNA in common, plus the measurement error to account for markers being a subset of the total DNA. So we want to solve for A, and we substitute A with G because G is, analogs to A. And the normalized method, again, the ZZ prime is weighted by the trace of the ZZ prime. So this method, make sure that, the outcome is compatible with A, especially when the inbreeding or the number of generation is low. So in the formula, N is the dimension of the matrix. And if the inbreeding is high, NB can be substituted with 1 plus the inbreeding coefficient. So the diagonal elements can be less than 1. So we obtain different, we use different methods to calculate the genomic relationships from the markers. There are some other methods. But one of the problems we run into when we obtain the genomic relationship matrix is getting the inverse. It can be singular, especially if the number of loci is limited. You have hundreds of markers, but thousands of individuals. Sometimes, two individuals have identical genotype across all the markers, and that can cause a problem. And sometimes the number of markers is smaller than the number of individuals genotype. And that can be a problem in taking the inverse of the G. So to avoid potential problems, the G matrix can be weighted as given in the formula. So here GR is unweighted genomic relationship matrix. A is the numerator relationship matrix among only genotyped animals. So it is actually a subset of A, and then W is the weight, and this is a constant actually. And Aguilar et al. Suggested that any number you type between 0.95 and 0.98, you get very similar results. So it's not really critical if you choose a constant between 0.95 and 0.98. All right, I think that's enough theory here, right? Let's go to R. I'm using RStudio for calculations. So first, we need to install genetic pad package from Bioconductor. And then we need to load the library, genetic pad library, to the memory so we can use it. So in case we have some other functions or objects in the workspace, we want to remove them using that rm command. Of course, we need to set the working directory. So first I'm defining the path and then setting the working directory. So, I can make sure that, I'm the directory I'm supposed to be. Yes, that's fine. So we are calling the source functions, which is the genomic rel.r function. And that's the one actually we are going to use to generate genomic relationships. Now, this is our marker table. This is a very small data sets. So we can click on the markers on the workspace and then to see the data. So the dimension of the marker, the data frame is 100 by 1001. So it's really small data set. And then we need the pedigree file. Okay. Pedigree file is just in a sparse format. We have individual IDs listed, their sire, and then now those are the five different methods we covered the theory to calculate genomic relationships, and GOF, GD, GO5, GMF, and then Greg method. I did not include normalized method, but I think that's enough to, show how we calculate genomic relationships. So I'm going through with each method. As you can see on the right side in the workspace, you can see those data frames are generated, matrices are generated. So regression method, takes a little bit longer. So if you click on one of those data frames, what you see is the column and row number for each individual to identify individuals, and then we have coefficients or elements in the matrix, in the genomic relationship matrix, which is G. But also we have the numerator relationship coefficients derives from the pedigree, which is A. So if the row equals to column or column equals to row, which means that's the diagonal element or the inbreeding coefficient. And if they are not, like, column is 2, row is 1, then we have a relationship coefficient, which is the off-diagonal element. Okay. Now we can, use the data frame and, combine all those matrices into one data frame. And then calculate correlations between them to see how they correlate with the A matrix. Okay, here are our correlations with A matrix. So this is the, the first column. If we look at those correlations with A matrix, GOF and Greg, which is a regression, produce very similar results. Also, GD produced very similar results. They are high; those are high correlations. GMF produced, the weakest correlation with the A matrix, which is 0.875. And then you see correlations with others, with each other. So we can plot like GOF and A. That shows the, how strong those two methods are correlated. This plot, of course, or correlations includes both diagonal inbreeding coefficients and off-diagonal elements. And we can look at distribution of the genomic relationship coefficients from GOF method based observed allele frequencies. Of course, we can take the subset the matrix or the data frame core OPT to look at the inbreeding, and then the relationship coefficients. or which are the off-diagonals. And then we can look at their maximum values, minimum values for each, and then the means. And then we can combine all those values, summary statistic in one data frame, and then visualize. And then mean I is the average inbreeding, and mean R is the average coefficient. So if you look at the mean I, which is the average inbreeding, A and Greg are exactly the same. GMF produce very high inbreeding coefficient compared to A matrix. Again, we can use plots and histograms to look at the relationships. So here we have relationship between off-diagonal elements of A and the genomic relationships Greg method, which is regression method. So, of course, you know, we need to obtain the inverse of one of those G matrices to use in the GBLUP. Here I am using the REL function to calculate the G inverse. And that may take some time, although this is a very small data set, you know, but for actual data sets, it can take some time. So it's done. Again, you know, we can view and then look at the dimensions. And then we can write the table, the inverse matrix for the genomic relationships in our working directory to use for genomic lab. Okay, let's go back to our slides. So if you look at the distribution of the genomic relationships derived from the markers, what you see as shown in that histogram, they are no longer discrete. You see a continuous distribution between 0 and 1. And most individuals have a coefficient of 0.5, which means they are full SIPs. But even among the full SIPs, you know, it's not always 0.5. You know, some SIPs, you know, share more genomes from the parents than other SIPs. That's why, you know, SIPs not exactly alike, except, you know, twins. So this is the major difference from additive genetic relationships derived from the pedigrees. Okay, let's move to traditional genetic evaluation using the pedigree and phenotype. This is just a review. I am sure everybody is somewhat familiar with that topic. This is just a reminder and then transition to genomic lab to see the differences. So we use linear mixed models to estimate breeding values of individuals. And we use phenotype and pedigrees. So in the mixed model, we have vector of observations. We have X and Z incidence matrices. X is associated with the fixed factor B, and Z is the incidence matrix for the random effects. And then we have an error term. So the major assumptions of the linear mixed models are expectations of the random effects and residuals equals to 0. And we assume random effects and residuals are independent of each other. And the variance of the random effects is the G matrix, which is the product of the additive genetic relationship matrix and the edigenetic variance. And then we assume that residuals have identity matrix, which is R matrix. You know, every individual has the same error variance, although those assumptions may change depending on the experimental design, especially for plant breeding. And, you know, to sum up, the variance of the observation is the product of the Z, G, and Z prime plus residual variance. So, and then we use mixed model equations to solve and to get the predictions for individuals. So the solutions for the fixed effects would be vector V, and then solutions for the random effects would be mu hat. So lambda is a ratio of residual variance and additive genetic variance, which is basically the inverse of the heritability. So Mendelian segregation effect, you know, when you don't have any phenotypic data for some individuals, what you get is mid-parent breeding value. They will be exactly the same for all the SIPs coming from across, because there is no data about those individuals, which means, you know, we cannot calculate Mendelian segregation effect. So what is the Mendelian segregation effect? When gametes are produced by meiosis, allele pairs separate, leaving each cell with a single allele. So sampling of parental alleles is random at each locus during the meiosis, and this is called Mendel's law of segregation. So each progeny receives about 50% of the parental DNA, and that 50% of DNA is randomly sampled. That's why, you know, SIPs do not look alike. So estimation of Mendelian sampling effect requires progeny phenotype. Or we can use markers to provide such information on which allele at a QTL was transmitted. So if we model the breeding value of an individual, it would be basically the summation of the mid-parent breeding values plus the Mendelian segregation effect plus an error term. So in the equation, mu sub j and mu sub k are the parental breeding values. Their average would be contribution to the individual breeding value. So it's large contribution. Genomic BLUP. So as we will see, genomic BLUP is relatively easy and really does not involve anything that we are not familiar with ABLUP. It's very easy to implement. All we need to do is, you know, replace the inverse of the A matrix derived from the pedigrees with the inverse of the G matrix derived from the markers to predict the breeding values. That's all. So this is a mixed model equation, a linear model for the genomic BLUP. Nothing is different, right? It's the same linear mixed model. One difference is that the Z in the model is the incidence matrix for marker effects. And the U is the vector of additive genetic effects that correspond to allelic substitution effects for each marker. So for each marker, we get a coefficient. And that coefficient represents the allelic substitution effect. And then we take the sum of all those coefficients across all the marker loci and then calculate the breeding values. So if I really simplify the genomic estimated breeding value for an individual, it's really a summation of all the marker effects in the model. Mixed model equations for the genomic BLUP, they are exactly the same, except lambda is different, and then we substitute A matrix inverse of the A with the inverse of the G, which is shown in red fonts. So lambda is different. It's defined as the sum across all the loci of the minor allele frequencies times the ratio of error and additive genetic variance. That's the same thing, you know, that ratio I'm talking about, inverse of the heritability. So, of course, in genomic BLUP, you know, some of the individuals may not have any phenotypic data. That's the idea. You know, we only have marker information. We don't have any phenotypic data, and we want to estimate their predictions or breeding values using the markers. And we, you know, accuracy of the genomic estimated breeding values will be different from accuracy of the breeding values of the individuals with some phenotypic data. So the first formula at the top, this is the accuracy of the breeding values for individuals with observation. So it's a product of the genetic matrix and error matrix and the variance components. For individuals without any observations, we substitute G with the C matrix, which represent genomic covariance matrix between individuals with and without observations. And C is obtained as weighting the ZZ prime with minor allele frequency, as shown in the corner, right corner. So fitting genomic BLUP, once we obtain G inverse matrix or inverse of the genomic relationship matrix, we can run the mixed model in any platform, in R or in SAS or in ASReml. This is you know, a model, a syntax for ASReml, for standalone ASReml. I think we have enough time for another demo, right, Sean? Can you confirm?
Can you hear me, Fikret?
Yes.
Yeah, we have a few more minutes, so you can, and still time for questions, so you can do another demo.
Okay. Okay, this is, you know, ASReml code. This is the data we have, the fields in the data, the tree effect, and then female and male of each tree we have, and they are associated with the pedigree. That's why we have P qualifier. And then we have other fields we are not interested in, and then we have the dependent variable, height and volume. So what is important here is that, you know, we need to have the pedigree file. This is our pedigree file here. And then we need to have the inverse of the genomic relationship matrix, G inverse. And then we need to list the data file. So we must have that order in order for program to run the genomic BLUP. So I have two parts in the code. The first part is classical BLUP using the pedigree and phenotypic data. And then here we have tree effect, which is the random effect, and that tree effect is associated with the inverse of the additive genetic relationship matrix. So we are using pedigree here, or inverse of the A matrix. This is error variance. It is fixed. And this is the additive genetic variance. Again, it is fixed because this is such a small data. I don't want to calculate variance components. I'm just fixing the variance components. And then at the bottom, we have genomic BLUP. The only difference is, substituting A inverse with the G inverse. That's all. Same model. So all I need to do, the program is going to run part 1 and part 2 consequently, and then rename the output for each part. That's what it does, rename. So part 1, ABLUP, part 2 is GBLUP, and all I need to do, run it. So this shows the scatter plot of the residuals from the ABLUP. And then this shows the scatter plot of the residuals from the genomic BLUP. So we have two different estimations, and we can use that data. We can use the different predictions and look at the correlations and look at the accuracy values, and then go from there. So we had demo already. So I have 3 more slides, and then maybe we can, have questions and answer session, okay? So this shows accuracy of predictions from genomic BLUP, and with predictions from traditional genetic evaluation, or ABLUP, we call it. So here in that paper published by Zapata Valenzuela, they split the population into training and validation, 2 different scenarios, and then they look at the correlations between predictions from ABLUP and GBLUP. And as you can see, genomic BLUP was even better than the traditional genetic evaluation. So the accuracy was 0.71 versus 0.6. And then in the second scenario, we got even better accuracies from genomic BLUP, because you are using more data, or the training population size is larger. So this scatter plot shows the correlations between predictions from ABLUP and genomic BLUP. In that scenario, we did not remove any phenotype from individuals. So just using the genomic relationship matrix instead of pedigrees to see what kind of correlation we get. So it's almost perfect correlation. Now this slide is important, and that's my last slide. So when you remove phenotype of subset of the individuals, if you use the traditional BLUP and pedigrees, what you see here is that, circled by red, those individuals are 6. They come from the same cross. So they don't have phenotypic data. The prediction will be exactly the same for all of them. That would be mid-parent breeding values. So you cannot make selections in the cross. But when you use markers or genomic BLUP, which is shown in the x-axis, you see segregation. They have different breeding values. They range from maybe 1 to 4, maybe 4.5. So markers allows capturing Mendelian segregation effect. And that's a very powerful tool to use in animal and plant breeding. So other advantages of genomic BLUP, especially for plant breeders, you can include experimental design factors in your mixed model, like environment replication effect. And it's very easy to run with various software. Some acknowledgements, those slides and material I have been using are taken from a textbook and some publications. The textbook is being developed by Chris Malteca, Jim Holland, Ross Wetton, and myself. And of course, we got a lot of help from our students, Jaime Zapata, Funda Öğüt, and I have to acknowledge NC State University Tree Improvement Program. With that, I'll be glad to take questions.